Hello and welcome. Now let's begin today's lecture. Previously, we mentioned the two most common approaches for obtaining raw NGS data. Those are obtaining raw NGS data from sequencing service providers or companies and downloading public raw NGS data from public databases. Let's see how we can obtain raw NGS data from sequencing service providers. It is quite straightforward. After sequencing is completed, the sequencing service provider usually delivers the raw sequencing data directly to the researcher. This is commonly done through email notifications, cloud storage links, FTP servers, or other file transfer systems. For example, here I have already downloaded the sequencing data files provided by the sequencing service provider. These are paired end FASTQ files including read1 or R1 and read2 or R2 file. These files were generated from paired end sequencing and were provided directly by the sequencing service provider after completion of the sequencing run. You can also notice that the files are provided in .fastq.gz format. The .gz indicates that the FASTQ files are compressed to reduce file size and make data transfer easier. We can extract or decompress these files to obtain raw FASTQ files for downstream bioinformatics analysis. As we can see here, this is the raw FASTQ file for read1 and this is the raw FASTQ file for read2. Now let's open this file. Here we can see the FASTQ entries. This is the first entry. This is the second FASTQ entry. And this is the third FASTQ entry. This particular dataset represents Illumina paired end 16S rRNA V3 to V4 Amplicon sequencing derived from a human stool sample. So this is one of the most common ways researchers obtain raw NGS sequencing data directly from sequencing facilities or commercial sequencing service providers. Similarly, we can also obtain and download raw shotgun metagenomics sequencing, whole genome sequencing, and RNA sequencing data from sequencing service providers via email, cloud storage links, or other file transfer systems. Now let's move to the second and one of the most widely used approaches, that is downloading public raw NGS data from public databases. Building on our previous discussion about SRA structure and accessions, we will now learn how to search and retrieve datasets from NCBI Sequence Read Archive or SRA database. Before downloading the NGS data, let's first see how to find relevant NGS datasets from the NCBI SRA database. Once we know how to search and identify the correct datasets, we can then move on to the downloading process step by step. Let's see how to perform a basic search in SRA. First, go to the NCBI Sequence Read Archive or SRA database. If you already know the address, you can directly copy and paste it into the browser or simply type the URL. Then press enter. This is the home page of the NCBI SRA database. Alternatively, you can simply search for NCBI SRA on Google and open the official link from there. Once the page opens, you will see the main search box as we can see here. This is where we can start exploring datasets. Basic search in SRA can be performed in different ways. One of the simplest and most straightforward method is search by accession. There are 4 hierarchical levels of SRA entries as we discussed in the previous lecture. These are bioproject or study, biosample, SRA experiment, and SRA run. If we already know the ID, We can search directly using the accession ID in this search box. Let's see an example. If we have a bioproject ID such as PRJNA1400113, so we can directly enter the bioproject ID as we have shown here and search it in SRA and access the associated bioproject data. Now click on search. Here we can see all the experiments associated with this bioproject. As we can see here, there are a total of 537 SRA experiments associated with this bioproject. If we click on or open any of these experiments, we can see the details of that experiment along with the associated metadata. Here we can see the details of that experiment along with the associated metadata. At the bottom of this page, we can find the SRA run ID and its associated information. As we can see here, this is the SRA run ID or accession. We can also directly access the bioproject page by selecting bioproject from the drop down menu before performing the search. Now click on search. Here we can see the bioproject page for this particular bioproject ID. Here we can see the details of this bioproject. Similarly, if we already know the SRA experiment ID, we can simply type the accession ID into the search box to directly access the corresponding experiment in the SRA database. For example, SRX324333665. Now click on search. Here we can see the details of this particular SRA experiment. Alternatively, we can also copy and paste the SRA experiment ID into the search box. Similarly, we can also search for biosample and SRA run records using their corresponding accession IDs. So by simply copying and pasting any of the accession IDs into the SRA search box, you can directly access the corresponding dataset at its exact hierarchical level. This is one of the fastest and most precise ways to retrieve specific sequencing data in the SRA database. Now let's move to another important method, search by words or phrases using NCBI's Entrance search engine. To search using keywords or words, simply type them into the search field. For example, 16S Amplicon human gut. Now click on search. Here we can see the results for this query. This type of search treats each keyword separately and returns results where these terms appear anywhere in the metadata or record. So you may get a wider range of datasets, including some that are not strictly 16S rRNA or not strictly human gut, but still contain overlapping keywords. To refine this further, we can use phrase searching. To search by exact word combination, enclose it in quotation mark. For example, here both 16S Amplicon and human gut are enclosed in quotation marks. Now let's use this refined query. Click on search. Here we can see the search result for this refined query. This tells Entrance to treat each quoted phrase as an exact match, improving specificity. It reduces unrelated results and help you retrieve more precise datasets where these exact phrases appear together in the metadata record. Now let's take this a step further with Boolean operators. You can modify your search results using logical or Boolean operators such as or, and not in your query. Or finds records that contain at least one of the terms. The second is and, which is the default operator in interest search, narrows the search by requiring all terms to be present. Finally, not, which excludes records that contain a specific term. Parentheses are used to build complex queries and control search logic. Now let's enter a query into the search box using these operators and parentheses. For example, and that is 16S rRNA and human gut. Next is or, then 16S Amplicon and fecal. We can use another parenthesis. Now we will use not cell line. This means the query will return results that contain either 16S rRNA and human gut or 16S Amplicon and fecal, but it will exclude any records that include the phrase cell line. You can also use Boolean operators in combination with accession numbers. You can also use wildcard characters during searching. Now click on search. Here we can see the search result for this query. Now let's move to the advanced searching using the SRA Advanced Search Builder. To begin advanced search in SRA, click on the link Advanced located just below the Universal Search field. This will open the Advanced Search Builder interface where we can apply multiple filters to refine our dataset selection. Now we will set the search fields using the default AND logic, which combines all conditions together. First, we can specify the search term or keyword such as human gut. We will keep the default all fields option unchanged as we can see here. Next, set the sequencing platform. From the drop-down menu, select platform. You can click on the show index list to view and select available platform options. Here we can see the list of available platforms. We can select Illumina. From the next drop-down menu, select the library strategy. As we can see here. Again, click on show index list to see available options. We can select Amplicon. After that, set the layout. From the next drop-down menu, select layout. Again, click on show index list. From the list, select paired. It is important to note that using too many strict filters in the SRA Advanced Search can significantly reduce the number of results. This is because metadata in public databases is often incomplete, inconsistent, or differently annotated across studies. Therefore, it is often better to start with broader filters, then gradually refine the search based on the results. Now click on search. Here we can see the search results for this query. So to summarize, In this lecture, we first demonstrated how raw NGS data can be obtained from sequencing service providers or companies. We then explored different ways to search the SRA database, including accession-based search, keyword-based search using entries, phrase searching using quotation marks, and Boolean operators for advanced query building. Finally, we introduced the advanced search builder in SRA and learned how to apply structured filters to refine dataset selection. In the next lecture, we will move to the practical section where we will learn how to download raw NGS data from the SRA database step by step. That's all for now. Thank you everyone.